Hello, and thank you for the people who are staying until the last presentation. I hope you win the iPads. And it is my honor to present research here at CFRI conference. And I have studied Pseudomonas aeruginosa since 2003, and CF always has been in my mind something that I need to study and be helpful. It's been 1 year since I got funded for this multi-omic study on concurrent sinus and lung disease in CF. The research is still ongoing, and I'll talk about what I have found so far. All right. So before I talk about the findings of our research, we need to talk a little bit about microbiome. The human microbiome study emerged about 15, 16 years ago, having the nickname that the bugs in your gut that makes you fat. They're probably the same bugs in my gut now. But since then, the microbiome study has come a long way. Now, many researchers beginning to understand its importance and the roles in various human diseases. The human microbiome studies discovered a surprising number of different microbes in a human and their functions in health. The term microbiome is the aggregated genome of all microbes in an environment, and the microbiota is the collection of microbes. So the microbes reside in the gut is the gut microbiota, and the genome of the gut microbiota is the gut microbiome. This studies revealed that we have about 1.3 times more human microbiota, the microbial cells in our body than our own cells. And there are approximately 100 times more genes encoded by the human microbiome than are encoded in the human genome in a person. And they encode for various functions in our body. And these microbes contribute not only in digestion, but also microbes in the gut aids the education of the immune system. And through interaction with our immune system, they contribute to health in peripheral organs such as lung and brain. So the changes in the microbiota can associate with diseases. And there is a remarkable diversity of human microbiota in health, and these communities have distinct composition across the human body. For example, the composition of GI microbiota is dominated by Bacteroidetes and Firmicutes, whereas the nostrils are dominated by Firmicutes, Actinobacteria, and to a lesser extent, the Proteobacteria. So in the airway, the microbiota burdens and the composition changes due to the health status. The microbial burdens of healthy individuals decrease from upper to lower airway. However, the microbial burdens of diseased airway were all high throughout the airways. And also the microbiota composition was different between healthy and diseased airways. The different diseases shows a different compositional patterns, and these observation leads to the hypothesis that the compositional changes of airway microbiota is related to the airway disease development. And among these different airway microbiome, my research is focused on the CRS and CF microbiome and their relationship with CF progression. Then what is CRS? The CRS is a type of sinusitis that approximately 14% of Western population suffers and costs about $65 billion per year for its management. And these are the pictures of sinuses. And one on the top is the sinus of a healthy person, and the lower two pictures are from the CRS patients. The left one shows the inflammation, and the right one shows the nasal polyp development in the sinus. In sinusitis, the nasal cavity is clogged by inflammation or polyps, and it gets filled with fluid, resulting in significant decrease in life quality. The sinusitis can be categorized into three different types based on the duration and frequency of the symptom. If the symptoms maintains over 12 weeks, it is called chronic rhinosinusitis. CRS. And CRS is clinically stratified by polyp presences. And despite the medicinal and surgical management, the recurrence of CRS is very common. Then why would we want to study CRS? First, the CRS is co-occur with CF very frequently. And there is a hypothesis called unified airway hypothesis, which postulates that a pathological process in one region of the airway would affect the function of the entire airway. There are epidemiologic and pathologic and clinical evidences which reveal the link between upper and lower airway diseases such as CRS and asthma, and CRS and COPD. And this recently published article shows the approximately 91% homology in the expressed genes between upper and lower airways under asthmatic condition. And Dr. Vaidyanathan briefly mentioned in today's talk about possibility of sinus being a reservoir for drug resistance microorganisms that may affect the pulmonary infections, which goes with the unified airway hypothesis. And in the unified airway hypothesis, microbiome is an emerging biomarker to study the hypothesis. But how about in CF? The progressive CF characterized by the presence of polymicrobial communities and chronic inflammation in tissue, sputa, and bowel fluid. However, there are only few studies that evaluate CF sinus and lung microbiome in parallel. The most of studies are focused on compositional features of the bacterial lung microbiota using 16S rRNA sequencing. But in our study, we are characterizing the microbiome using shallow shotgun metagenomics in order to identify microorganisms that contribute both to upper and lower airway health or disease in CF patients. Let's look at the previous studies we've done, and these graphs shows the upper airway microbiome dysbiosis in CRS patients with and without asthma and CF. Here, we can see the microbial community richness decreased the most in CRS patients with CF. And this graph shows the diversity analysis of sinus and lung microbiome of CRS patients with or without CF. It shows the loss of microbial diversity in both sinuses and lungs of CRS patients with CF compared to the CRS patients without CF. And these indicates the loss of microbial niche specificity between sinus and lung in CF patients, which means the microbiota of sinus and lung of CF patients became similar. And this is the unbiased hierarchical clustering of CF sinus and lung samples with microbial community composition. And sinus and lung samples from individual patients are generally confined to statistically distinct clusters. And exceptions include patients following lung transplant and with a typical CF mutation, both of which are characterized by increased diversity of lung specimen. And these previous results shows the substantial bacterial community collapse in the sinuses and a loss of niche specificity between the sinus and lung when compared to non-CF patients with CRS, which supports the unified airway hypothesis too. And this is the first specific aim for our research. We will use comparative metagenomics in banked lung and sinus samples from healthy CRS and CF-CRS patients collected at UCSF. And we will compare these strain level taxonomy features to identify the microbial components that co-occurs in sinus and lung disease in CF-CRS patients. The inclusion of lung transplant patients will allow us to infer the directionality of microbial translocation in this subset of patients. You are probably thinking, this is so boring, and what is it talking about? A good thing about Zoom presentation is I can't see your facial expression or you're sleeping or not. But for the people who are still awake, let me just restate what I was saying. There are tons of microorganisms organisms living in airway, and these dwellers affect their surroundings, sometimes in a good way and sometimes in a harmful way. It is like human on Earth. In this research, I'm trying to find who these dwellers are and what they do to the airways, and hopefully find someone, the microbes, who can make this word, airways, better. All right, so this is the demographics of some of our participants. We have banked about approximately 700 pairs sinuses and lung specimens from 246 CRS patients with varying comorbidities such as CF and healthy individuals for controls. Sample collection is still ongoing, and we are also investigating the association with clinical data, including SNOT20 for CRS and recent antibiotic use or recent steroid use on the microbiome. The main difference of our research with previous studies is the use of metagenomics approach. The past researchers used 16S sequencing, which only targets 16S rRNA genes of bacteria, or ITS sequencing for fungus only. But shotgun sequencing sequences all given genomic DNA from a sample, and shotgun sequencing achieves higher resolution to strain level compared to 16S or ITS sequencing, which yields species level at its best, and is often utilized when functional profile is required, which is the part of our study. We have decided to use shallow shotgun sequencing approach since the coverage and the resolution of shotgun and shallow shotgun sequencing for human microbiome related samples are almost same. There are several different analysis platforms for the shallow shotgun sequencing. In order to select the best analysis platform for and the condition, and we did some preliminary studies on several samples, and this is MGREST analysis of right bronchus sample of a CF patient. MGREST assigned taxonomy to sequences, but the resolution was lower than what we expected. And the functional pathway analysis using KEGG showed the majority of genes associated with microbial metabolism. And another platform for Kraken-2, problem was Kraken-2, and Kraken-2 is a valuable tool for metagenomics taxonomic analysis of individual samples. And shotgun analysis using Kraken-2 provides the most detailed microbial compositional analysis, including eukarya, bacteria, archaea, and viruses, yielding an average of 6,000 taxonomic hits per sample, and times 700, that's how much we have. However, we can see here the lack of visualization tool, and group analysis tools makes analysis between groups very difficult. There is a visualization tool for Kraken-2 called Corona, but Corona had a limitation on the microbes it can show, maybe because it's based on the environmental analysis, and is still an individual sample-based analysis. So in order to overcome this weakness of Kraken-2, we have incorporated the bioinformatics tool called QIIME2 analysis. And this is taxonomic bar plot presenting relative abundance of composition of bacterial microbiome. And I want to thank Dr. Hoffman for explaining the bar plot and the PCOA plots so nicely in the previous talk. The bacterial compositional analysis demonstrated, and expectedly, the CF patients had a higher relative abundance of Pseudomonas aeruginosa compared to non-CF samples. However, our novel approach to understand the upper and lower airway microbiota have characterized these taxa to the strain level and allowing us to investigate these communities at a higher resolution. And interestingly, the CF patients and CRS with polyposis patients had co-occurrence of Acinetobacter baumannii in their microbiome, the yellow bars. And using CHIME2, we calculated the beta diversity of evaluated changes to evaluate changes in diversity of composition across clinical states. We demonstrated a compositional shift by clinical comorbidity using Bray-Curtis and Jacquard dissimilarity metrics. And there was no obvious clustering between controls and CRS samples, but CF samples, those red dots, presented clustering apart from the other groups, which means the CF samples have different bacterial dwellers compared to the other groups. We also calculated the alpha diversity, it's a richness, using observed OTUs, group significance, and Shannon diversity index using CHIME2. In the bacterial microbiome, the richness between body sites, sinus and lungs, and clinical states were similar, except one significant increase in observed OTUs in CF compared to CRS specimen. Since we included samples from lung transplant patients, this was somewhat expected. And now something new to the field. We were also able to evaluate the fungal microbiome using Kraken-2 and CHIME2. The reason why we evaluated the fungal microbiome is because we have shown that there are interesting interactions between bacteria and fungus in sinonasal cavity of mice that affect the microbial composition. And this is very fresh out of the oven, and I just got accepted for publication last Friday. And back to the slide, we did not demonstrate considerable difference in their composition between CF and CRS patients, but there are definite different fungal members exist in only diseased samples compared to healthy samples. So we could observe Botrytis cinerea, Collectotrichum higansianum, Yarrowia lipolytica, Candida dubliniensis. It's mouthful. And several other fungal species in lesser abundance, only in CRS and CF patients, both lung and sinus, but not in the healthy sample. And the beta diversity analysis shows changes in diversity of or composition across clinical states. Both metrics presented distinct two clusters, and only one of those clusters contains healthy controls, which is in purple dots. Also, the pairwise Parmenova statistical analysis shows that there is significant dissimilarity between healthy samples and CF or CRS samples. And the alpha diversity analysis of the fungal microbiome revealed that there is no significant difference among body sites, but it showed significantly higher qualitative and quantitative richness in CRS and CF microbiome than the healthy samples. This was quite interesting when you think of bacterial microbiome commonly loses their richness when the disease occurs. We'll definitely look into this more in the upcoming studies. And in order to determine the concordance between CS16S RNA gene sequencing approaches to evaluate the microbiota and shallow shotgun metagenomics, we compared the microbiome profile of several overlapping samples that had been sequenced using both approaches. The dominant members of the microbiome were matched with the result of the shotgun metagenomics analysis, which was of Pseudomonas aeruginosa. However, there were some mismatched results among low abundance members, such as like Acromonas, or Acromobacter or Stenotrophomonas. And this could be due to the difference in reference database used in each analysis. And this can be resolved when we develop a reference database compatible to both analysis and is currently in progress. But we observed similar compositional shift by Bray-Curtis and Jacquard's dissimilarity analysis by clinical status, showing the separation between CF and CRS org controls. But In the alpha diversity analysis of bacterial microbiome using 16S rRNA sequencing demonstrated different results compared to shallow shotgun analysis. The qualitative community richness is decreased in CF patients with or without incorporation of phylogenetic relationship between features. And this could be due to the lower taxonomic resolution of 16S rRNA rRNA sequencing analysis. For example, like when 16S rRNA analysis identify one species of Pseudomonas aeruginosa, the shallow shotgun sequencing identified 16 different strains of Pseudomonas aeruginosa, which brings those richness up in shallow shotgun sequencing. So in this research, I have to say summary, not conclusion, because it's not been concluded. And we found that Kraken-2 provide the most detailed microbial compositional analysis, including eukarya, bacteria, and archaea and viruses. And in bacterial microbiome analysis, we found or confirmed the CF patients had a higher relative abundance of Pseudomonas aeruginosa compared to non-CF samples. And CF patients and CRS with polyposis patients had co-occurrence of Acinetobacter baumannii in their microbiome. We could also see compositional shift in the sinonasal and lung community by clinical status. And in fungal microbiome, we could see those unique fungal members in diseased samples. And we could see Botrytis cinerea, Collectotrichum higginsianum, Yarovia lipolytica, and Candida dubliniensis. All right, yeah, and only CRS and CF patients, but not in the healthy controls. And we could also see the compositional shift between CF or CRS and control. And these preliminary findings demonstrate the advantage of the metagenomics approach to resolve taxa to the strain level and to include fungal, viral, and archaea microbiota community members in understanding the ecology of the airway microbiota. And we could also found that the metagenomic sequencing has a higher dependence on the reference database. And there was difference on the results between 16S rRNA or ITS sequencing analysis and metagenomics analysis, possibly due to the difference in reference database used in each analysis. So we are currently developing a reference database compatible to both analyses. And hopefully I'll be back next year with the specific AIM2 results, which will be the function of the microbial members, those members that we just saw, hopefully, the Acinetobacter baumannii and all the fungal members, and their roles in host immune response in CF. And these are the great people and organizations who made all this possible. Thank you for listening.